overall goal of this protocol is to synthesize non-chemically cross-linked foams, or microcarriers, derived from tissue-specific extracellular matrix for applications in 3D in vitro cell culture models, or as pro-regenerative scaffolds in cell delivery systems.
This method can help answer key questions in the fields of applied cell biology and tissue engineering, such as how the 3D cellular microenvironment mediates cell function and tissue regeneration.
The main advantage of this technique is that it enables the fabrication of scaffolds with tunable geometries that mimic the native tissue-specific ECM composition and are stable in long-term culture without additional cross-linking.
After lyophilizing the tissue-derived ECM according to the text protocol, use sharp surgical scissors to finely mince the decellularized tissue. To cryomill the processed tissue, fill a 25 milliliter stainless steel milling chamber for a laboratory ball mill system with minced lyophilized ECM, and add two 10 millimeter stainless steel milling balls. Close and completely submerge the loaded milling chamber in liquid nitrogen for 3 minutes. Then mill the frozen sample at 30 Hz for 3 minutes. Repeat the submersion in liquid nitrogen and the milling until the ECM is milled into a fine powder. Weigh out 250 milligrams of either minced or cryo-milled ECM and transfer it to a 15 milliliter centrifuge tube. Add 5 milliliters of 0.22 molar NaH2PO4 buffer